eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to synthesize messenger RNA or pre-mRNA with the help of enzyme RNA polymerase.
The process of transcription occurs in three stages, which are initiation stage, elongation stage, and termination stage.
The enzyme that synthesizes RNA from the DNA template during transcription is RNA polymerase. There are three types of RNA polymerase in eukaryotic cell, and RNA polymerase II is responsible for transcribing protein coding gene.
There are 6 general transcription factors denoted as TF2D, TF2A, TF2B, TF2F, TF2E, and TF2H. They are responsible for promoter recognition and for unwinding the promoter DNA.
Enhancer and silencer are the two elements which are important in transcription. Enhancers increase gene promoter activity, while silencers repress gene activity.
Two other important eukaryotic polymerases are RNA Polymerase I and RNA Polymerase III. RNA Polymerase I resides in nucleus for large ribosomal RNA precursor synthesis, while RNA Polymerase III is responsible for synthesis of tRNA, rRNA, and some snRNAs.
In initiation stage, there are promoters at the start site, RNA coding gene, and the terminator at the stop site. In promoter, data box is the source of RNA synthesis. Firstly, TF2D with TBP binds to the data box. TBP DNA complex provides a platform to recruit other transcription factors and the polymerase to the promoter. The binding followed by TF2A, TF2B, TF2F with polymerase, then TF2E and TF2H. This is called pre-initiation complex. The wiggly tail consists of protein complexes carried by RNA polymerase II, which are CPSF and CSTF.
Once RNA polymerase II has escaped the promoter, the enzyme polymerase moves along the DNA template downstream from 3' to 5' end, shedding most of the initiation factors. Meanwhile, RNA chain is elongated in 5' to 3' direction. RNA polymerase II locally unwound the DNA double helix for RNA synthesis. One of the exposed DNA strands acts as the template for RNA synthesis using nucleotide by the ribonucleoside triphosphate (NTPs). Guanine is added to this 3' to 5' end by an enzyme called methyltransferase at position 7 to give rise to 7-methylguanine cap. The cap is protecting the 5' end from 5' exonuclease. RNA polymerase II synthesizes the pre-mRNA chain until it reaches the termination site.
Once the sequence is transcribed into RNA, it will trigger transfer of CPSF and CSTF to RNA, which are called as poly-A signals. After the binding of the protein complexes to RNA, other proteins are recruited elsewhere, leading initially to RNA cleavage and then polyadenylation of the 3' end of RNA. The overhanging RNA is digested by a 5' exonuclease, which is XRN2 protein. It helps the RNA polymerase II to disengage from its DNA template strand, finally terminating that round of transcription.
Pre-mRNA is the strand of RNA that is made during transcription. It consists of 5' cap and 3' poly-A tail. The poly-A polymerase enzyme catalyzes the addition of 3' poly-A tail that forms with CPSF and CSTF. Basically, there are three differences between the transcription of RNA polymerase in eukaryotic and prokaryotic cell. The first one is eukaryotic promoters are more varied due to the multiple forms of RNA polymerase with different specialization. Also, the binding of eukaryotic RNA polymerase to DNA template require the participation of transcription factors, while not in prokaryotes. Plus, protein-to-protein interaction also occur in eukaryote while not happen in prokaryote.